Well, hey there. The results are back. It looks like you have sarcoidosis.
Sarcoidosis? What in the world is that?
Sarcoidosis is generally a systemic inflammatory disease characterized by the presence of granulomas in any organ.
Wow. That sounds scary. Can you break down more for me what that means?
Of course, let's start by discussing some key parts within the immune system so you can understand how they play a role within sarcoidosis. Your innate immune system is your body's first line of defense against pathogens and begins acting within hours to days. Key components of the innate immune system include cytokines, PAMPs, PRRs, macrophages, and dendritic cells. Cytokines are small soluble proteins used to communicate between cells that can regulate the development or activation of target cells. Important cytokines in sarcoidosis include IL-6, 10, 17, 18, TNF-alpha, and IFN-gamma. IL-6 and TNF-alpha are cytokines that induce a state of inflammation at the site of infection. IL-18 is also a pro-inflammatory cytokine that has the ability to induce development of Th1 cells. IL-17 induces the production of granulocyte colony-stimulating factors. IFN-gamma activates macrophages and Th1 cells. IL-10 has potent anti-inflammatory properties that play a central role in limiting host immune responses. Another important component of innate immunity are pathogen-associated molecular patterns, or PAMPs. PAMPs are small molecular motifs conserved between multiple microbes. PAMPs are recognized by innate immune cells through the use of pathogen recognition receptors, or PRRs. An important PRR that is involved in sarcoidosis is TLR2. Toll-like receptor 2, or TLR2, allows for the production of TNF-alpha when activated. In sarcoidosis, TLR2s have a single nucleotide polymorphism that leads to enhanced responses from activated TLR2s and therefore increased amounts of TNF-alpha production. Interestingly, TLR2 is also induced by TNF-alpha, which creates a positive feedback loop that enhances inflammation as well as the granulomatous creation that is characteristic of sarcoidosis. Macrophages are initially derived from monocytes that arrive from the blood. The main function of macrophages is to recognize, phagocytize, and kill pathogens, as well as raising awareness to other immune cells an infection is present. In sarcoidosis, macrophages do not work as efficiently, leaving behind poorly degraded antigens. Macrophages can either be activated classically or alternatively. Classical activation of macrophages results in phagocytosis and promotion of the immune response, as we have already discussed. However, when macrophages are alternatively activated, they create tissue fibrosis, which is seen in sarcoidosis. Dendritic cells are known to take up extracellular antigens when their PRRs are activated by PAMPs. When activated, dendritic cells travel to the lymph node to activate T cells by presenting an antigen on an MHC molecule. Both dendritic cells and macrophages are capable of presenting antigens via MHC molecules to T cells and are collectively referred to as antigen-presenting cells. Within the disease pathology of sarcoidosis, MHC molecules on APCs are upregulated, which in turn impacts the activation of the T cells. Now let's talk about aspects of the adaptive immunity. Your adaptive immune system represents your body's ability to mount an attack against specific foreign invaders. The adaptive immune system can be split into cellular immunity involving T cells and humoral immunity involving B cells. B cells develop and mature in the bone marrow and travel through the blood to the lymph node where they mature and are activated when a pathogen is present. B cells express B cell receptors that recognize and bind antigen. When activated, B cells can differentiate into plasma cells which produce antibodies. B cells also have the ability to differentiate into memory B cells after activation, which reside in the lymph node and can be reactivated if infection occurs again. In sarcoidosis, patients are seen to have decreased levels of memory B cells. In normal immunity, B cells also are seen to secrete cytokine IL-10, which I mentioned earlier is responsible for regulating the immune response via anti-inflammatory mechanisms. In sarcoidosis, B cells secrete less IL-10, which contributes to an uncontrolled immune response and the development of fibrosis. T cells also create T cell receptors that recognize protein-based antigens that are presented to the T cells via MHC molecules. There are two types of MHC molecules that interact with T cells. MHC class I molecules are found on the surface of all your cells and interact with CD8 positive T cells or cytotoxic T cells. MHC class I molecules present to T cells both self-antigens and antigens from infections in the cell cytosol. MHC class II molecules are only found on antigen-presenting cells and present phagocytosed antigens to CD4 plus T cells or helper T cells. In sarcoidosis, deficiencies are seen in the number and function of cytotoxic T cells, making it more difficult for the adaptive immune system to eradicate infected cells. Helper T cells also have the capability to differentiate into different subtypes based on the cytokines present to help better fight the infection at hand. Th1 cells are used to fight intracellular infections, further classically activate macrophages, and secrete IFN gamma that activates other helper T cells to become Th1. In sarcoidosis, Th1 transcription factors are upregulated, which also upregulates cytokine secretion through these pathways. Chronic and increased cytokine stimulation contributes to the creation of fibrosis and granulomas that is seen in sarcoidosis. Th17 cells fight microbial extracellular infections, activate neutrophils, and secrete IL-17. Now let's discuss some of the unique elements involved in sarcoidosis. The primary etiology of sarcoidosis revolves around the creation of a granulomatous inflammation response. This results from persistent infection due to poorly degraded antigens and is maintained through the continual recruitment of macrophages and T cells to the site of the granuloma. The persistence of antigens and immunostimulatory molecules contribute to the inflammation that creates the pathology of sarcoidosis. Granulomas are small lumps of immune cells that form in areas of infection. In sarcoidosis, granulomas are non-necrotizing, meaning the tissue is still alive, create an environment with antigen remnants, and end up altering organ structure and function. Other factors that contribute to sarcoidosis are serum amyloid A and BAFF. Serum amyloid A are proteins normally used to transport immune cells to inflammatory sites, but in sarcoidosis is seen to accumulate within granulomas. BAFF is a cell survival signal found in sarcoidosis that binds B-cell membrane receptors that inhibits apoptosis and stimulates the differentiation of B cells into plasma cells. BAFF levels are seen to be increased in sarcoidosis patients, which contributes to the prolonged state of inflammation. Okay, let's start putting all the pieces of sarcoidosis together now. Sarcoidosis is initially initiated by either environmental factors alone or environmental factors interacting with genetic factors that initiate the innate immune response. It is hypothesized that sarcoidosis is initiated by an altered composition of lung or gut microbiota that may act as a source of PAMPs. In sarcoidosis, macrophages are unable to eliminate the infection through phagocytosis, which leads to pathogen containment. Major cytokines released by the macrophages are IL-6, 12, and 18. These cytokines then act on the mTORC pathway, which is responsible for inducing differentiation of activated macrophages with contained pathogens into epithelioid cells. Epithelioid cells are non-necrotizing granulomas that exhibit some degree of lymphocytic inflammation with the release of TNF-alpha, which promotes the formation of sarcoid granulomas. Released cytokines also initiate an acute phase response by upregulating SAA within the body in an attempt to create an inhospitable environment for the infection at hand. Increased levels of SAA then begin to accumulate within the granulomas and promote enhanced T cell responses. With an increased T cell response to antigen, while under the influence of the cytokines and dendritic cells, T helper cells are directed to polarize into the subsets Th1 and Th17. The activated Th1 and 17 cells further promote inflammatory processes through the release of cytokines, which contribute to granuloma formation. Macrophages at the site of infection that have yet to differentiate into epithelial cells continue to secrete chemokines, which attract more Th1 and 17 cells and monocytes. While all of this is occurring, there are also depleted levels of IL-10 being secreted by B cells, which contributes to the uncontrolled immune response and development of fibrosis. The continued, unregulated, and enhanced effects of the macrophage and effector T cell responses result in a sarcoidosis epithelial granulomatous creation. Hope that better helps you understand sarcoidosis.